And I will go ahead and just kick this off by introducing this session, which is music, building bridges in the brain. And I think after you watch the symposium, you'll see why we called it that. I just wanted to quickly introduce Dr. Emmeline Edwards, who is the director of the Division of Extramural Research at the National Center for Complementary and Integrative Health, also known as NCCIH. She, in her earlier life, got her PhD in neurochemistry and did a lot of behavioral pharmacology. She is currently the chair of the Women in the World Neuroscience and as an independent mentoring and networking organization, and really helps with implementing mentoring and networking opportunities for women. With that, I would like to introduce Dr. Edwards to get us started. Thank you.
Good afternoon, everyone, and also welcome to our virtual attendees. So this afternoon, we have a very interesting symposium for you, a focus that the NIH has been really involved in for the past 7 years. So I want to thank my collaborators in putting this symposium together, Dr. Wen Chen, who is the program director at NCCIH, and also Dr. Dana Schlosser, who is the program director in the Office of Behavioral and Social Sciences. So our panelists today, I'm going to give you a little bit of context for the talks that are going to follow, but then we have Dr. Maria Geffen from University of Pennsylvania, Dr. Kevin Liu from the intramural program at NIDCR and NCCIH, Dr. Melinda McPherson from the University of California, San Diego, and Dr. Narayan Sankaran from the University of California, Berkeley. I have no conflict of interest as a federal employee. So as a way of introduction, I want to tell you a bit about the premise for music-based intervention, tell you a little bit about the state of the music and health research field and the efforts that we've been involved in at the NIH. But most important for this audience, I want to tell you about what further research is needed and how we can get there. So about 7 years ago, Dr. Francis Collin at the time was the NIH director, and he started a collaboration with a very famous soprano, Renee Fleming, and both of them charged us to actually develop a symposium at a workshop at the NIH, which was the first time that we had brought together neuroscientists and music therapists and advocacy group representative. And at this workshop, we actually talked about where we were in the context of how music impacts the brain and how that could be used as potential therapeutic interventions. And so as a result of this workshop in 2017, we actually had a paper that was published in Neuron in 2018. And out of that, we had a set of recommendations that we've been really implementing from that point on. So the first thing that we started was to develop a number of funding opportunities to allow us to do some research in the context of both basic and mechanistic research, as well as clinical and translational research in the field of music and health. And to date, we have, the NIH really has had a fairly large investment of about $40 million in that endeavor. So this is an opportunity for Brain's initiative, PIs, to move into this direction, which is very exciting. So the work that has been funded so far really is already confirming that musical experience really has a profound effect, not only on enhancing the brain and cognitive development, but also in improving function and well-being, as well as optimizing quality of life and possibly ameliorate the symptoms of a broad range of disease and disorders. During the COVID, pandemic, we actually utilized that time to have 3 very large virtual workshops, where we work with interdisciplinary groups of neuroscientists, music therapists, advocacy group, clinical trialists, and also behavioral researchers to put together a number of initiatives. One of them was to really develop, to improve the methods by which investigators were carrying out music-based intervention. So the first workshop focused primarily on how to do a rigorous music-based intervention. The second workshop focused primarily on what kind of outcome measures would be beneficial to look at, especially in the context of brain disorders of aging. And the third one was what kind of biomarkers would also be necessary to. And earlier last year, we actually have a paper published in Neurology, which is really the NIH music-based intervention toolkit, which is really a set of guidelines to the community of how to do this kind of research. Currently, we have a funding opportunity that's actually focused on testing the guiding principle of the toolkit. So this is going to be open through October. So if anyone is interested, this is really to carry out feasibility trials of the music-based intervention toolkit, especially in the context of brain disorders of aging. The next endeavor that we got involved with is really to build this community of researchers. And this is why this symposium is so important, because we want to bring researchers that are funded by the Brain Initiative to this kind of research, because we're interested in understanding the neural circuits that are activated by music. So we want to engage a group of interdisciplinary researchers. And we certainly want to have a cross-training of neuroscientists and music professionals to carry out that kind of research. So the way that we're accomplishing this is by actually currently funding a number of high-priority research networks. We currently have 4 of them, and they are focused on music interventions. But the primary focus is to better understand the mechanism by which music-based intervention exert their effect. And we're also interested in technologies, tools, and models, which is why this symposium is relevant. So this is the 4 networks that we have funded. 3 of them are focused on music and pain, and 1 on music and Alzheimer's. And those networks are meant to actually bring communities together, so they are open. So if anybody wants to collaborate, you can contact us and we'll put you in contact with the investigators. In December of last year, we had a very exciting 2-day workshop called Music as Medicine, the Science and Clinical Practice. And we, as a result of this 2-day meeting, we have a comment in Lancet, which was published this past March, really outlining what are some of the challenges and also the needed action. And one of the needed action, as you can see, is to incorporate music-related hypotheses and data collection into major national initiatives such as the Brain Initiative, hence this symposium. So one of the things that I want to highlight is that we know some of the circuitry that's involved, but as you can see, the auditory pathway is connected to the motor system, the cognitive system, the affective and motivational system, and also other sensory systems. So there's a lot of work to be done. Today we're going to hear about 2 of our investigators are working in rodent models, and they're going to be talking about the corticocollicular circuit, and a second one is going to talk about the cortico-thalamic circuits. And then the last 2 speakers are going to talk about their work with human subjects and focusing on different aspects of the musical components. So again, we do need further studies with different models. And certainly technology is really important as to how to carry this work and to actually demonstrate the potential of music-based intervention. So without further ado, I'm going to let Dr. Maria Geffen from the University of Pennsylvania tell us about her work. Maria?
Thank you, Evelyn. I'm thrilled to be here. Thanks so much for inviting me to be part of this symposium. Today, I'm going to tell you about recent work from our lab on identifying the neuronal circuits for detecting sound patterns. If you were now at a musical concert, listening to this pianist play a piece by Rachmaninoff, your brain would be performing a number of auditory computations. For one such computation, and perhaps the most fundamental one, is discrimination of sound frequency. If you think of the piano, each individual note produces sound at different frequencies. As you move up or down the piano, the frequency increases or decreases, and as the pianist hits different musical keys, your brain is comparing the frequency produced by that sound. Another computation that's really important for your perception of music is identifying sequences of notes. And that's the first step in detecting and identifying music melodies. Part of that process of identifying sequences of tones is identifying the statistical structure in those sequences and making predictions for what the next note is that's going to come. That process is fundamental, not just to music perception, but also to all other forms of auditory communication in navigation. In my laboratory, we have been studying the neuronal circuits for auditory perception and learning, focusing on the central auditory pathway. In particular, our goal has been to bridge the gap between the understanding how individual neurons encode and represent information and how our brain makes sense of sounds and produces behavioral and emotional responses. In order to do that, we have focused on the intermediate level, that of neuronal ensembles. And our focus has been on understanding at the cellular level and at the level of projections between different brain regions what the mechanisms are for various neuronal computations that make up complex auditory processing. In understanding how the brain processes sequences of notes, we have focused first on the very simple sequences. Imagine that you have two tones. Imagine you're pressing two notes, but one of those tones is presented to you much more frequently than the other note. If we record the activity in the primary auditory cortex, which is the key region in the brain that transforms the representation of a sound waveform into an object-based representation, almost all neurons in that brain region will exhibit a diminished response to the tone that has been that's presented more frequently, and an enhanced response, a stronger response to the tone that's presented less frequently, that's unexpected tone. And this does not depend on it's not specific to the frequency of those two respective tones. If we switch them around and now the higher frequency tone is presented more frequently and the lower frequency tone is presented more rarely, this relationship flips and the neuron becomes more responsive to the lower frequency tone than the higher frequency tone. This process is termed stimulus-specific adaptation. Within the auditory system, we encounter stimulus-specific adaptation within the central pathway, starting at the inferior colliculus, traveling up to the medial geniculate body and up to the auditory cortex. But the auditory cortex sends extensive massive feedback to the thalamus and to the inferior colliculus. And so a question that we have been trying to answer is, how is stimulus-specific adaptation determined mechanistically and implemented mechanistically within the auditory cortex, and what are the signals that are transmitted down to the inferior colliculus with respect to the prediction and statistical expectation. If we compare the signals to the standard tone and the deviant tone within the ascending auditory pathway between the inferior colliculus, the auditory thalamus, and the auditory cortex, the difference increases. Stimulus-specific adaptation is really strongest in the primary auditory cortex within the ascending auditory pathway. And my lab has shown with our previous studies that stimulus-specific adaptation relies on the interactions between excitatory and inhibitory neurons within the cortex. The mammalian cortex is comprised of excitatory and inhibitory neurons, which form intricate circuits. But there is a pattern to this madness of inhibitory-excitatory interactions, with some neurons, somatostatin neurons, targeting the distal dendrites of excitatory neurons, whereas other neurons, parvalbumin-positive inhibitory neurons, targeting the cell bodies. We were able to show that it's specifically the somatostatin-positive inhibitory neurons within the primary auditory cortex that contribute to stimulus-specific adaptation. Well, what about the signal that travels down to the inferior colliculus? We became interested in what aspects of stimulus-specific adaptation is actually transmitted back to the earlier stages in the auditory pathway. If we look more closely at this process of the reduction of signal to the repeated tone and the enhancement of signal to the unexpected, the deviant tone, we can actually separate out stimulus-specific adaptation into two separate processes: repetition suppression and calculation representation of prediction error. And that allows us to tie this analysis to the predictive coding framework, which postulates that neural networks learn the statistical regularities inherent to the natural world. And as part of the processing, the predictable components in representations of the sensory inputs are removed. And here, this can happen through repetition suppression. Whereas what the brain tries to transmit to the upstream or downstream target are prediction errors. In order to be able to separate out these components from stimulus-specific adaptation, we can present another stimulus, which is a cascade stimulus. To those of you who have spent their childhood practicing scales, you will quickly recognize this stimulus. In fact, for many of you, this might be really overrepresented in your brain. Luckily, our mice are more naive, and that is not a confound in the study, at least not one that we considered. If we present, but in this cascade stimulus, we can present the same tones, but in a perfectly predictable fashion. And we can compare the responses to the standard tone in the audible stimulus to the same tone in the cascade stimulus in order to isolate the repetition suppression component. And we can compare the responses to the deviant tone to the same tone presented in the cascade stimulus in order to isolate the prediction error component of the response. It has previously been shown that within the auditory pathway, again, the prediction error is also strongest in the auditory cortex. However, if we look more closely in the inferior colliculus, which is actually comprised on multiple regions, the central region, which is part of the lemniscal pathway, the ascending pathway, and the shell of the inferior colliculus, which is the region that receives feedback from the auditory cortex. The prediction error is actually stronger represented in the shell. So the postdoc in my lab, Alex Lesico, who has amazingly recently been awarded the Brain Initiative K99. Just wanted to throw it out there. We're very grateful. Ask this question: how does the input from the auditory cortex affect predictive coding in the inferior colliculus? In order to do that, she recorded the neuronal activity from the shell and central regions of the inferior colliculus in awake, hat-fixed mice presented with the audible stimulus, and inactivated the feedback from the auditory cortex to the inferior colliculus using optogenetic methods, and compared the changes in the prediction error and the repetition suppression components with and within the inferior colliculus, with and without corticocollicular feedback. I would not be standing here in front of you today if she did not discover that corticocollicular inactivation in fact caused bidirectional changes and affected both the changes in response in the standard and the deviant context. But more specifically, she found that the corticocollicular inactivation reduced prediction error in both regions, and it did not affect the repetition error as strongly, and it did not affect it significantly in the central region of the inferior colliculus, suggesting that the corticocollicular feedback is selectively transmitting the signal predominantly about the prediction error. Now, interestingly, when we investigated these signals more closely, we found that whereas some neurons indeed exhibited classical stimulus-specific adaptation and prediction error, there was quite a number of neurons that in fact were facilitated by the presentations of the same tone, which we termed the repetition enhancement. And in fact, the responses to the deviant tone were reduced, almost in the mirror of the error error signal that you would expect, which we call the negative prediction error. We did find that corticocollicular inactivation also changed this negative response metrics. So the corticocollicular inactivation selectively reduced repetition enhancement in the central nucleus, but the negative prediction error was modulated in the shell of the inferior colliculus, so in the feedback receptive regions. So combined, the study found that predictive signals are transmitted via the corticocollicular feedback from the auditory cortex to regions earlier in the central auditory pathway. Now, the study points to potential targets for investigating how our brain represents complex sequences of music, complete sequences of sounds, such as are found in music. And our laboratory is currently investigating how these excitatory-inhibitory interactions and the feedback between different regions affect pattern detection in behaving mice. I would like to thank and acknowledge that this was largely the work of Alex Lesico, a postdoc in the lab, and I would like to thank all of our lab members and our sources of funding, and in particular the Brain Initiative, which makes research on the systems neuroscience possible.
Thank you much. I'm going to ask that we hold the question to the end because we do have a Q&A session. So let's move on to our next speaker, Dr. Kevin Liu. Kevin?
Thanks a lot. Thanks a lot for Emily for organizing such great event, which providing me the opportunity to share some recent findings. So a long-standing observation is that certain sounds, ranging from the diverse genres of music to even sounds of nature, induce pain relief in humans. So our study was inspired by a clinical observation published more than 50 years ago. In that case, by observing a cohort of 5,000 dental operations, a group of dentists in Boston documented that playing music during dental operation could reduce pain. So interestingly, they documented that it wasn't just the music itself, but even noises, as we can imagine, that was generated during dental operations could have analgesic effect. So inspired by this clinical observation, we sought out to look at whether and how music or sound could produce analgesic effect using rodent models. To start, we first established an inflammation pain model in mice. And then we performed behavioral tests using the von Frey filament, in which punctate mechanical stimuli of different pressures were delivered onto the hypoplantar surface so that we can assess their mechanical sensitivity. As we can see in this video, I'm sorry, not music yet. After the mice, the inflammation pain was generated, when we only applied the low force von Frey filament, which is normally not painful at all, mouse now develop robust high force flinching and leaking. Both are indicators of mechanical pain hypersensitivity. So when we confirmed this, then we added a music during the von Frey test. So the music I chose is a piece of Bach symphony. And we can see now, sorry, this video. And I hope you can appreciate now with this Bach music, the same low force von Frey stimuli, which is quite painful, no longer evoked hypoflinching, suggesting that the mechanical pain hypersensitivity was reduced. So when we saw this, of course, we played with different conditions. So it turns out that such analgesic effects are highly dependent on the relative intensities of the music that was played over the background noise. For example, when the ambient noise was set as 45 decibel, so we discovered that only the music was played at 50 decibel, but not a high level intensity could generate analgesic effect. In addition, we discovered that when mice were received repeated exposure to music for 3 days, then such analgesic effect could last for additional 2 days, even without playing music during the test. So we were really curious what behind drives this, music-induced analgesia. Was it due to the music itself? For example, mice really loved Bach, but not Mozart or Beethoven. By the way, why we chose Bach is because my graduate students believe that Bach music had the best, it has best harmony, which I respectively disagree because I believe Mozart has more beautiful melody. But who knows how mouse will respond. Or it was due to other properties of sound. So to test this, we played different music. So the first, we switched pitches of this Bach music, converted from a consonant music to dissonant music, so that it sounds quite unpleasant to human beings. So it's the same music of Bach, but we just switch pitches. So it doesn't make any sense to humans. And then we also play them with white noise background. No music, just sound. And then test them for the von Frey test. So as we can see that both conditions achieved quite similar level of analgesic effects. So we were really surprised. So what was the key? So we discovered that in all three conditions, what in common is that as long as the music or sound is played at a level that is slightly above the ambient noise, in this case, 5 decibel above the background noise, it could drive analgesic effects. So the key here is really the low signal-to-noise ratio sound, but not music per se, at least for mice that were tested. So we then went further to investigate how does such low signal-to-noise ratio sound could drive analgesic effects. To do that, we first probed neural activity within the auditory cortex, where all sound information is processed. So by performing in vivo multielectrode recording of the auditory cortex, we did discover that only the low signal-to-noise ratio, I'm so sorry about that, only the low signal-to-noise ratio sound, but not the high ones, could reduce the spontaneous activity within the auditory cortex. So when we performed chemogenetic silencing of the excitatory neurons within auditory cortex, we discovered that the inhibition of excitatory auditory cortical neurons could significantly elevate the reduced mechanical threshold seen after inflammation. These results suggest that the auditory cortex is actively involved in the low signal-to-noise ratio sound-induced analgesia. We then went further to identify the potential downstream targets that might mediate such cross-modality sensory processing. To do that, we injected pre-dependent synaptrophin mRuby into the auditory cortex of Calcamus 2 Cree mice. So this method allows us to specifically target the presynaptic terminal of different afferents derived from the auditory cortex of those excitatory neurons. So to our surprise, besides projecting to the auditory thalamus, which is known so that I don't show here, we identified abundant mRuby signals within the somatosensory thalamic nucleus, including the thalamic posterior nucleus, the PO, and the ventral posterior nucleus, the VP. So these two nuclei in the thalamus are known as somatosensory thalamic nuclei. They receive and relay the ascending information, including both innocuous and nociceptive, transmitted from the spinal cord, and then send them to the somatosensory cortex. So when we see this structure, we hypothesize that there is the structure to function correlation, which means these somatosensory thalamus might function as a bridge for audio somatosensory processing. We then went further to focus on the functional dissection of such auditory cortical to somatosensory thalamic pathway that mediate sound-induced analgesia. And for today's talk, I will mainly focus on one that projects to the PO. So to start, again, we performed in vivo multielectrode electrical recordings within the PO. We discovered that under inflammation pain conditions, the spontaneous neural activity was significantly enhanced within PO. And quite interestingly, such neural overactivation can be sufficiently reduced when mice were exposure to the low signal-to-noise ratio sound. To further probe the function of this auditory cortical to somatosensory thalamic pathway, we performed optogenetic stimulation. So to do that, we expressed different options in excited neurons within the auditory cortex, and then placed a fiber on top of different somatosensory thalamic nuclei, in this case PO. So by shedding different wavelengths of light, this allows us to specifically target the axon terminals derived from the auditory cortex. So we start with optogenetic inhibition. As shown here, optogenetic inhibit of this pathway did reduce the spontaneous activity within the PO. And moreover, it elevated the significantly reduced mechanical threshold after inflammation. Therefore, inhibiting this pathway phenocopied the low threshold signal-to-noise ratio sound exposure. And we then went further to perform optogenetic activation. It turns out that if we activate this pathway, it could block the low threshold sound, the low signal-to-noise ratio sound-induced analgesia. So suggesting that this auditory cortical to somatosensory thalamic pathway is both sufficient and required to drive sound-induced analgesia. So I would summarize my talk here. Our findings did discover that the auditory cortex play a crucial role in affecting the nociceptive processing by impacting the cortical-thalamic cortical transmission that is required for somatosensory nociceptive processing. Of course, there are a lot of remaining questions that deserve to be further studied. It's hard to believe that the cortical thalamic pathway is the only one that might drive sound-induced analgesia. So we are also very interested in the cortical-cortical connections. In recent human studies, these connections are suggested to play an important role in music-induced analgesia, particularly the insular cortex that receive convergent input both from the somatosensory cortex and high limbar cortical regions. And we are also interested in other subcortical pathways. For example, at the brainstem, the supracolliculus, which integrates multiple somatosensory inputs. So with this, I'll stop here and acknowledge my lab. And great thanks to our wonderful collaborator, Dr. Zhang in University of Science and Technology in China. And particularly the first author, Wenjie, who agreed to join my lab before the pandemic, but cannot come. So this is really a wonderful cross-continent collaboration for this cross-modality sensory processing. Thank you.
Dr. Melinda McPherson is going to have a presentation on diversity in pitch perception. Melinda?
Where's the next?
Oh, just see.
Okay. All right. Perfect.
And what is the red?
Okay. Great. Wonderful. It's a pleasure to be here today to tell you about some of my recent work exploring the psychology and the neuroscience of pitch perception. So pitch is one of the most salient dimensions of all of the sounds we hear around us every day. So for example, when we're identifying environmental sounds like computer chimes, bird songs, train whistles, we can often characterize those sounds based on their pitch. Pitch also conveys critical information in speech, where it can tell us the difference between a question and a statement, like this.
You know what I mean. You know what I mean? Me. Me. See you tomorrow. See you tomorrow.
And of course, most critically to the symposium today, pitch conveys critical information in music. So sequences of pitches give us melody, and pitches at the same time will offer us harmony. So today I'll be telling you about some new advances we have made in understanding the psychology and the neuroscience of pitch perception, and how we've been reevaluating classic theories of pitch. So sounds said to have pitch are generated by periodic or repetitive processes in the world. So for example, the vibration of a violin string or a guitar string as pictured here, the vibration of a speaker membrane, or the vibration of your vocal cords as you speak. And the rate of repetition of those periodic sounds is referred to as the fundamental frequency, or F0. So for example, if you have a waveform like this pictured here that repeats 200 times a second, it's produced by a periodic process in the world that vibrates 200 times a second, it has a fundamental frequency, or F0, of 200 hertz. So what happens to be true is that sounds that are periodic in time are generally harmonic. So they have this structure in frequency. These harmonics are integer ratios of a common fundamental frequency, or F0. So what I'm showing here on the screen is a spectrogram, which is a time frequency decomposition of sound. On the y-axis we have frequency, on the x-axis we have time. And this is a representation of a singer just singing an ascending and descending note. And you can see these bands of energy, those black lines. So those are the harmonics, and again, each harmonic is an integer ratio of this common fundamental frequency, or F0. So pitch is generally defined, or has classically been defined psychoacoustically, as our percept, which corresponds to this F0 and this periodicity and harmonicity in the world. So what's classically been thought is that pitch representation or pitch discrimination is assumed to rely on representations of the F0. So if you're asked to do a classic pitch discrimination task where you hear two tones, it's thought that what you'll do is you'll estimate this fundamental frequency of the first tone, estimate the fundamental frequency of the second tone, and then compare those two estimates. However, listeners could use other representations to perform this computation to detect that one tone is higher than the other. So for example, you could just track that each of these harmonics shifts concurrently, so that all these harmonics are shifting together. So recently, I've been working on this question and trying to re-examine how we actually do this task. I've been working on looking at what is actually the function of F0-based pitch, and actually do we have potentially multiple pitch systems. So to do this, I've been taking harmonic stimuli like this, sort of the standard pitch types of stimuli that we'll hear all around us, and I've made them inharmonic. And I've done this by jittering the harmonic frequencies to make them inconsistent with any single fundamental frequency. So these new inharmonic stimuli have no consistent pattern in harmonic space and frequency space, and they are no longer periodic in time. The logic of this approach is that tasks that are thought to rely on a representation of the fundamental frequency should be greatly impaired with these inharmonic stimuli. So I've taken these harmonic and these inharmonic stimuli and I've run a basic discrimination task, which has been done hundreds of prior times. It's a sort of the classic task for diagnosing pitch perception, where you ask participants, is the second note higher or lower than the first? And according to most traditional models of pitch perception, this task should be greatly impaired. Performance on this task should be impaired when tones are inharmonic. But I'll play you an example trial, and hopefully you'll be able to hear that the second note is higher than the first. See if I can play that again. Okay. So when we ran this experiment, we found something that we didn't quite expect. So I'll orient you on this axis. On the y-axis we have percent correct, and on the x-axis here we have difference in semitones. So the pitch difference between the two notes that were presented to listeners increases as we go from left to right. And critically, what we found was comparable performance for harmonic in blue and inharmonic stimulus in red. So this suggests that F0 is evidently not necessary for discriminating basic up-down movements in pitch. We ran some follow-up control conditions that indicate that what listeners are actually doing in this inharmonic case is just tracking that all these harmonics move concurrently. So critical to our inharmonic manipulation was that we actually modified the harmonics in each of the two notes in the same way. So the pattern of inharmonicity remained consistent between the two notes, and listeners were able to, what seemed like trivially or spontaneously, just track this pattern. So to reiterate, classic definitions equate pitch to representations of F0, but it seems like basic pitch discrimination, this basic task, actually doesn't require a representation of the F0 or fundamental frequency. It turns out that pitch, this basic feature of music perception, is potentially subserved by several different underlying mechanisms. And it presented to me at least a really interesting puzzle. So what actually is pitch perception? Does it rely on multiple underlying systems? So in order to gain more clarity on this, on the role of harmonic structure in pitch perception, I took this basic up-down pitch discrimination task, and I extended it by just adding a silent gap between the two notes. My question was, does introducing a delay make discrimination worse for inharmonic compared to harmonic tones? So I wanted to add a memory load here because memory is actually a really critical part of music perception. It's often critical to remember one note over the course of a melody in order to understand the musical structure. I'll show you the results of this experiment on this axis here. So on the y-axis here, we have a discrimination threshold. So here, lower is better, so we were measuring just noticeable difference in pitch. And on the x-axis, we have delay, so 0 versus 3 seconds. And here's what we found when we ran pitch discrimination thresholds for harmonic and inharmonic stimuli. So first, on the left, you can see that we had comparable pitch discrimination thresholds for harmonic and inharmonic stimuli, replicating our previous effect that I just showed you. However, when we added even a short delay of 3 seconds, discrimination of the inharmonic tones was considerably worse than that for harmonic tones. So this result is suggestive of memory representations that are somehow specific to harmonic sounds. It suggests again that what people classically call pitch, this harmonic structure, actually might be functioning for memory. So to review, in these inharmonic conditions, listeners must use a representation of the spectrum. They sort of track that all the harmonics move up and down concurrently. In the harmonic case, listeners could track the spectrum. It's consistent from note to note. They could also track the F0. And so we actually wanted to test a third condition, which we called interleaved harmonic, where what we did is we actually had no overlapping harmonics from note to note. So we'd take half the harmonics in the first note, half the harmonics in the second note. They were still integer multiples of a common fundamental frequency. And so our assumption in this interleaved harmonic condition was that people must use a representation of the F0 in order to discriminate that the two tones had changed. So I'll play you an example of this stimulus, and hopefully you'll hear that the second note is higher than the first. One more time. And when we ran these pitch discrimination thresholds, we found two key results. So first, we overall found that performance is worse in this interleaved harmonic condition with no delay. But critically, we found that there was no significant effect of delay on this condition. So the results suggest that harmonic sounds are overall better remembered, even though the spectrum might be used for back-to-back pitch changes. So this provided pretty compelling circumstantial evidence that what listeners are actually doing here is transitioning between two separate pitch systems, one where they track the spectrum and one where they track the F0. But again, this was circumstantial evidence, so we sought to find more concrete evidence. And to do this, we leveraged this memory effect and an individual differences approach. So we measured pitch discrimination thresholds in many people, with and without delays, and the logic is that performance on tasks that rely on the same underlying representations should correlate across participants. So here's a design of this experiment. Again, we use these three stimuli, harmonic, inharmonic, and interleaved harmonic. We tested pitch discrimination thresholds with and without delays. I'll be showing you the correlation in performance across those conditions on this axis on the right here. But I'll just quickly go through a prediction. So let's imagine in this harmonic case, listeners are using a representation of the fundamental frequency to do the task, and in the interleaved harmonic case, they're also using a representation of the F0. We can maybe imagine that every participant is using these same two strategies for these two different conditions. What we would expect to find is that performance on these tasks, if they are relying on the same underlying representation, should be correlated. And so therefore, we might expect to see a result like this, where there's a high correlation between these two conditions across both no delay and a delay. However, this is not what we found. Instead, we found a significant interaction between the correlations between harmonic and inharmonic, and harmonic and interleaved harmonic. So we found overall that, we found overall that listeners had there was a strong correlation in performance between harmonic and inharmonic stimuli without a delay, and this decreased with a delay. And then we saw the opposite pattern with our two harmonic conditions. We saw an increase with the delay. And so this result suggests that listeners are actually switching between two different pitch codes depending on the memory demands of the task. And I'll just emphasize that we ran this experiment actually using online data collection, and so we were able to easily replicate it using a slightly different paradigm. So we replicated it, the replication is in the bottom left, in an additional almost 200 participants, and found the same basic effect. So this effect is robust. Okay, so this effect does to suggest that memory for pitch requires a representation of the F0, but real-time pitch perception requires a representation of the spectrum, and people seem to fluidly move between these two modes of listening. So more recently, I've been interested in how this is instantiated in the brain. So I've been investigating neural responses to inharmonic tones using fMRI. And so what we've done is we've had listeners just sit in the scanner and listen to melodies made up of harmonic tones, inharmonic tones, and then noise that's spectrally matched to those tones. We've taken half of our data and tried to identify pitch-sensitive voxels. So voxels respond more to harmonic tones than spectrally matched noise, which is a classic contrast used to identify these pitch-sensitive voxels in auditory cortex. And once we've identified these pitch-sensitive voxels, we've taken the other half of our data and looked at the average response of those specific voxels to our different conditions. When we do this, we found our percent signal change plotted on the left here for both harmonic and inharmonic conditions is comparable. We actually find very similar responses in these pitch-sensitive voxels to inharmonic tones. This suggests that tracking the spectrum might be really a basic aspect of pitch perception for real-world sounds, and this is represented well in primary auditory cortex. So today I've shown you evidence for two different pitch representations. It seems like memory for pitch, again, is reliant on representations of F0. However, real-time pitch perception requires these representations of the spectrum. And I've also shown you that pitch regions of auditory cortex do respond similarly to harmonic and inharmonic sounds. I've actually used this basic contrast between harmonic and inharmonic stimuli as a basis for a much broader investigation of the role of harmonic structure in hearing. And I found that harmonic structure, or F0, seems to be critical not only for remembering pitch, like I've shown you, but musical interval discrimination and out-of-key note detection, hearing and discriminating tones and noise, and also voice recognition. However, this basic aspect of pitch, F0-based pitch, doesn't actually seem to be necessary for the classic psychoacoustic assessment of pitch, basic pitch discrimination. It doesn't help provide invariance to timbral differences, so the differences between different instruments that you might hear in music, or it doesn't seem to be important, or critical at least, for speech prosody and lexical tone discrimination, for example, Mandarin tone discrimination. So hopefully I've convinced you today that pitch representations are richer and much more interesting than previously thought. I think there's a great scope here for future work to look at how these multiple pitch systems are instantiated in the brain, how this relates to animal models of pitch and hearing, and also how pitch perception is modified by different disease states, and how we can maybe optimize assisted hearing devices like cochlear implants in order to more accurately represent pitch. I'd like to thank you all for listening. I'd like to thank the NIDCD for their funding, my former labs, the Lab for Computational Audition and the Vision and Memory Lab, and if you're interested in this work, I'll be continuing it in my own lab this fall at Purdue University. Thank you all for listening.
Our last speaker is Dr. Narayan Sankaran. He's going to talk to us about the encoding of melody in human auditory cortex. Narayan?
Okay. Thank you, everyone. Thanks to the organizers of this symposium. I'm thrilled to be here to talk about some work we've been doing recently. Okay, so at a very broad level, we're interested in understanding how a simple sequence of tones arrives at the ears and then gets mapped or transformed into meaningful and often emotion-inducing representations of melody. So within this process, we have the STG, which is a hub for complex sound processing that we've long known is implicated in the processing of melody. But beyond the simple question of whether the STG is active when we're listening, we really understand very little about the specific information that's being encoded in this region. And so in the work I'll present to you today, we had two main questions we were evaluating. So first, we simply asked what features of melody are encoded by neural populations in the STG, and what is the neural code by which these features map onto the neural activity. And then secondly, we asked whether this encoding that we're seeing occurs via mechanisms that are specialized for music alone, or whether it really makes use of these general purpose sound encoding mechanisms that might also be shared across speech. And so the method we used for this is ECOG. So these are direct high density recordings made from the temporal lobe in humans. And importantly, for each electrode, we're examining the high frequency band of activity. So that's between 70 and 150 hertz. Okay, so while we're recording ECOG activity, each of our subjects were presented with a set of natural instrumental melodies, about 25 minutes long. And in turn, this was comprised of about 200 different individual melodies, each about 6 seconds long on average. Okay, so before examining tuning to any specific features, here we're just visualizing the activation pattern across the STG in response to one melody. And what I'm sure you can all see is during the course of melodic processing, there's this very complex and spatiotemporally dynamic activation broadly across the STG. And so now the really important question is, how do perceptually important features of melody or aspects of melody map onto these activation patterns that we're seeing? And so in order to answer that, we need to know a little bit about what those perceptually important features are. So let's just consider a simple melody. Hopefully you can hear this. Okay, so I'm sure a salient feature you all heard is the discrete pitch that's associated with each tone. As you've probably heard by now, this is really the fundamental dimension along which melody varies in addition to time. But we can also consider the pitch changes that occur between adjacent notes in melody, and this is what we measure in semitones. And independent of the absolute pitch, it's actually these pitch changes that underlie our recognition of a given melody. And so while these two different properties, pitch and pitch change, are important for perception or recognition, we of course also have an aesthetic or an emotional response to music. And to consider these sort of higher order processes, we need to look beyond these short-term acoustic features. And so as Maria was describing earlier, we can use a pre-trained statistical model of music to measure how unexpected each note in the melody was, conditioned on the notes that came before it. And the key idea here being that, melodic sequences aren't random, but instead they have these statistical rules that govern how they're likely to progress. So in this melody, for example, the note in the position that's indicated by the red arrow is relatively unexpected because it's violating the pattern that was established earlier in the phrase. So it's a fair question to ask, well, why should we care about this feature? Well, it turns out that as listeners, we have an implicit knowledge of these statistical rules, and our emotional response to melody is thought to be directly tied to the extent to which these expectations are met or denied as we listen. Okay, so we have these three really perceptually important features of melody: pitch, pitch change, and expectation, and we want to examine how they're encoded in that neural population activity. And in particular, we were really interested in evaluating this really fundamental question, which is, are these three different features jointly encoded by the same neural populations, or are they each kind of independently encoded by separate populations? Okay, so here I'm showing you the response at a single electrode, and we've time-aligned the activity so that time 0 corresponds to the onset of notes. So we identified electrodes such as this one in which responses were clearly modulated by pitch. So in this case, you can see that this electrode's tuned to relatively low values of pitch, but we can see that the activity is relatively unmodulated across these contrasting levels of pitch change and expectation. So in a different set of STG electrodes, such as the one in the second column, we found tuning to pitch change, but not to the other two features. So you can see that this particular electrode really prefers descending pitch changes in melody. And then finally, interestingly, we found electrodes like this one that were positively modulated by how unexpected a note is, but not by the pitch or the pitch change of those notes. So really, taken together, what these response patterns suggest is that different properties of melody are encoded by independent neural populations. And so to formally test this, we used encoding models where we're essentially trying to predict the music-evoked activity from a set of different stimulus features. And then we can calculate the unique variance explained by each of our features of interest: pitch, pitch change, and expectation. And this allows us to essentially quantify the encoding of each of these three features at every electrode. And when we do this, we find that all three features are encoded in the STG across this kind of spatial map that you can see here. And consistent with those single electrode responses I just showed you, we see that for the most part, each electrode's activity is explained by just a singular feature of melody. This is also pretty evident if we look at the weights of the model for just 3 example electrodes, where we can see that for each electrode, which are these columns here, you can see that the weights are primarily concentrated over a single feature. So this is just one subject's data, but if we can essentially see the same result if we pool electrodes across participants and then directly compare the encoding of 2 given features. So let's just focus on this rightmost panel here. You can see that these kind of pink electrodes distributed along the x-axis are significantly tuned to pitch change, and these are orthogonal from these dark green electrodes that are tuned to expectation along the y-axis. So our first important result is that these 3 perceptually critical features of melody, despite all essentially being computed from pitch, are each selectively encoded within these separate populations in the auditory cortex. So we also characterize the neural code for each feature, or the specific format in which this stimulus information maps onto neural activity. But in the interest of time, I won't go into this response tuning. So if you're interested in this, come find me later, or I'll refer you to our recent publication down here. Okay, so having established how melodic information is encoded in this higher order region of the auditory cortex, we can now turn to the question of whether this encoding is specialized for music. And recently, research has shown that there's these music-selective populations in the auditory cortex. So there are neural populations that selectively respond to music and not other sounds. But so far, it's really remained a mystery what specifically is driving the activity on those music selective populations. So what information about music is being represented? And we had a very specific hypothesis about this. We thought, okay, well, pitch and pitch change are general acoustic features, but expectation really describes the unique statistical structure of music. So perhaps it's the encoding of expectation that is systematically driving activity in music selective populations. And so what we did is we played speech to the same ECOG participants. And here on the scatter plot, I'm just comparing the magnitude of the response to music versus speech. And really importantly, we found a small subset of electrodes that are quite strongly selective for music. So it's these blue electrodes above the diagonal. And if we just zoom in on one of those electrodes, we can verify that this truly is a music selective population. You can see in this individual trial raster that it responds to every melody in our stimulus, and it consistently doesn't respond to all the speech tokens. And so now the key question is, how do electrodes such as this one relate to those that encoded expectation? And so in this scatter plot on the right, the axes are exactly the same as in the leftmost panel, but the appearance of each electrode now indicates how strongly it was tuned to expectation. And what you can clearly see is those electrodes that are tuned to expectation are the ones that are music selective. And in fact, even when we control for the encoding of all the other variables, only the extent of expectation encoding can predict the degree to which a population is selective for music. And so together, these results really support the hypothesis that the encoding of expectation is a specialization for music. Now, even though we found that the encoding of these acoustic variables are not predictive of music selectivity, it's worth noting how acoustically different music and speech are. So if we just look at these time frequency representations, you can see that there's fundamentally different spectrotemporal structures. And so a worry that we had is maybe the music selectivity we're seeing doesn't reflect expectation. Maybe it simply reflects tuning to the acoustic structure of music. And so to control for this, we presented subjects with a third stimulus that we call melodic speech. So this is acoustically almost identical to speech, but we just altered the pitch slightly by discretizing its value within each syllable to the nearest musical note. So now we effectively transform these sentences into melodies. So here is an example of 3 different sentences. First, I'll just play you the regular speech version.
This is a sleeping capsule.
No temptation, no virtue.
Maybe it would work the other way.
Okay, now I'll play the altered melodic speech version, which I've just primed with a piano chord.
This is a sleeping capsule.
No temptation, no virtue.
Maybe it would work the other way.
Okay, so with very little acoustic perturbation, we're able to kind of create coherent melodies from speech. And the crux of the findings are, when we look at music selective electrodes, such as this one, which hardly responded to speech, we see a clear response to the melodic speech stimulus, despite its physical similarity with speech. So now we can more confidently say that the music selectivity we were previously seeing isn't simply driven by low-level acoustics. Instead, we can retain the physical structure of speech, and if we insert expectations into that structure, we see a selective response emerge. Just one more important point to note is that prior studies really found that music-specific activity clustered in these distinct regions of the auditory cortex. Contrary to that, we really didn't find much evidence for a music region. You can see that music-selective electrodes are kind of broadly distributed throughout the SDG and kind of intermixed with other sound-responsive populations. Okay, and then finally, in contrast to expectation, we hypothesized that these lower order acoustic variables of melody are domain general representations shared with speech. And the short of it is that's exactly what we found. So here I'm just showing results for pitch change. And this correlation indicates that the same set of electrodes are tuned to pitch change across both music and speech. And moreover, if we visualize the actual response tuning of these electrodes, we see that there's a highly similar pattern of tuning across music, which is the top matrix, and speech, which is on the bottom. So not only is pitch change being encoded by the same populations, but the neural coding of that information seems to be highly shared across domains. Okay, so to conclude, the kind of model for the encoding of melody that we arrive at is we see that it's really multidimensional in nature and makes use of both domain-specific and domain-general mechanisms. So the encoding of expectation, or the statistical structure of melody, is a specialization in the human brain for music. But the encoding of pitch and pitch change recruit these general purpose mechanisms that represent the equivalent properties in speech. So I'll stop there, and I'd just like to acknowledge my co-authors who were my mentors for this work that I did while I was a postdoc in the Chang Lab at UCSF. Thank you.
So thank you very much to our speakers for those wonderful talks. So we now have about 20 minutes of a moderated discussion. As we mentioned earlier, there's a lot of gaps and potential opportunities. We still need to better understand the impact of specific musical components. When we do a music-based intervention, we really don't know the difference between having a receptive or an active, process, whether or not you can be just listening to music or being engaged actively, whether individual intervention versus group intervention are different, and very relevant to this audience, really what new technology would allow us to better track brain function. So I'm going to open it up for questions right now. Let me just first ask our speakers if they have any questions for each other. If not, anybody from the audience?
Thank you all for your talks.
Okay.
Really appreciate it. We had close to 70 people join us online, and I have a few questions. I'll start with one. This is for Dr. Sankaran. Do this encoding and general observations, are they influenced by musical training?
Okay, so this work is done in a very specific patient population because it's using ECOG. So we weren't able to look at musical training in the work I mentioned. I would assume that the one feature of interest that could vary with musical training is expectation, because at least some of that is really reflecting a learned model. So the more we're exposed to the statistical structure of music throughout our lifetime, the stronger those internal representations of expectation. So yeah, musical training presumably enhances that.
Also for Dr. Sankaran, it's fascinating that this circuit for expectation seems to be there, and it probably suggests it's been there a long time. So do you want to speculate what this says about the evolutionary benefit of being able to perceive a change in pitch that surprises you?
Gosh. Well, I think it's definitely something that's adaptive for survival. The question of why it would be music specific is more confusing to answer and difficult to even speculate on at this stage. Whether it's evolved from more primitive mechanisms that allowed us to, I don't know, detect prosody and stuff like that in speech, that's one possibility. But I don't know, it's very difficult to speculate.
There are people who think maybe music preceded language.
I would actually jump in and argue that, mechanisms for predictive coding for detecting statistical regularities in structure and building predictions is so fundamental to perception that I think actually music has, evolved or been constructed to take advantage of our brain's ability to detect those, to form those predictive signals.
Yeah, I think this question is for Melinda. I was quite interested in the matching of fundamental frequencies versus detection just of relative changes. And it suggests that there's a template by which you would be matching this. So there's been a lot of discussion amongst musicians about whether you can develop perfect pitch or whether that's something that is just encoded from the beginning. And I wondered if you might respond to that idea. Where is that template stored? And if it is a template, a memory of some sort, can it be acquired?
That's a great question. I will just emphasize we weren't trying to match performance. It just, they ended up looking incredibly comparable when, no matter how we measured the pitch discrimination thresholds. So there has been a lot of work looking at whether we have these internal templates for this harmonic structure. We independently jittered each of those harmonics for every single new trial, and people are able to just spontaneously track that. So it doesn't seem like we have any kind of, at least from this data, concrete template that these notes are just sort of slotting into, because again, they are random every time. It seems like we have a more potentially general mechanism for just for tracking that these harmonics are concurrent and are congruently moving together, whether or not they fit into any standard harmonic structure. So, and it does seem that happens spontaneously, as you heard in that one trial. You sort of just hear it. So I don't think there's a concrete template there that people are following. I think it's more of an overall approach to listening to these types of sounds, almost like a movement detection.
Cool, awesome panel. My question's for Dr. Sankaran. I was really interested, you didn't go into too much details, but it looked like your stimulus set had a lot of different musical instruments, and then your speech data set had a lot of different speakers. One of the dimensions of music that you didn't really get into too much is, are there any electrodes that encode sort of sound source, whether that's musical instruments or different speakers, or if those overlap at all? It seems like you've got the data set to figure it out.
The short answer is yes, there are. So this is something we're just beginning to unpack. The representation of timbre and spectrotemporal variations independent of the melodic features I talked about in the STG. And certainly there's a lot of information there that we're just beginning to characterize.
I have a question for Dr. Sankaran and Dr. Liu. For Dr. Sankaran, for expectation, let's say you play the George Gershwin little excerpt there. If you're familiar with it, then why is expectation violated? If you had prior knowledge of a given piece, why wouldn't that be reflected? So do you find a reduction in firing of the cells that are, surprised by the lack of predictability once you become familiar?
Yeah, again, hard to say because in our patients, they reported being unfamiliar with the vast majority of melodies. But there is some work on why so-called veridical expectations, when we're familiar with a piece, why they're still encoded by the brain and why they're still important for example, eliciting musical emotions. Some of it is to do with, a dissociation between conscious processing of expectations and what is almost a preconscious or obligatory processing of a violation of a pattern.
Okay. Yeah, I mean, it sounds also as if your musical speech is very much like motherese or parentese. It's kind of intriguing that you find such a increase in response. For Dr. Liu, I'm still struggling to understand why you get the result that you do for the reduction of pain, the analgesic effect. I mean, you have a 5 dB SNR difference. Presumably, you get more firing for cells if there was a larger stimulus, right? So why is it that you have this profound effect with this very small discrimination between background and sound? Can you offer an explanation of it? And related to that, suppose you had the animal or the human engaged in attention to sound. So would that also reduce the response to pain?
Yes. I can first start with the second one. So I think in the field, it's always debating music or sound, right? If it produces an analgesic effect, does it due to it just brings your attention, not the sound per se? So that's a very good question. So we cannot rule out that, but as I introduced the data, if the mice received a repeated exposure for 3 days. Then we tested them again. The analgesic effect lasts for additional 2 days. And during that test condition, no sound, no music. They were not exposed to any sound or music. That suggests that it's not, primarily due to the attention, because during that test conditions, they are just themselves, no music, sound exposure. And for the first question, why the 5 decibel is a key? That's a great question and a very challenging question. We did test different signal to noise ratio. I mean, relatively to the ambient noise. I wouldn't say the 5, because technically we cannot achieve lower, because you can imagine the ambient noise is just fluctuating. So the 5 decibel is minimally we can accurately control. So then we also test slightly above like 10 decibel and gradually increase to 15 to 20. And we did discover that such very small differences between the sound and the ambient noise is most effective. There are some studies not in the pain field that just explaining like why animal is perceiving small sound above the ambient noise. They'll have some innate defensive behaviors, and that actually evolutionary that might make sense so that they get alert. But a lot of innate defensive behavior is coupled with pain reduction, because evolutionally they're just coupled. I hope that helped.
Right, thank you.
I had a question for Dr. Sankaran and then Dr. Liu. For Dr. Sankaran, I was curious if the recording that you made for expectation neurons, were they on the left or the right side of the brain? And is there any asymmetry that you see with respect to that?
Yeah. So no significant lateralization of any of the features. And we had pretty good sampling in both hemispheres. So I know this is contrary to some prior reports, but we really don't find strong right hemisphere lateralization for any of the melodic features.
Good to know. And for Dr. Liu, I was just curious, when you played Bach to the mice, then did you see any activation in the pleasure centers of the brain?
That's a great question. We have a look at pleasure centers. I assume that might be actually for mice. It's I don't know if there is a pleasant center for music, like how mouse perceive music or what is the music to mouse. We don't know. But in human studies, people did discover that different music, it's quite case by case. For individual person, everyone has a different taste. But there are a lot of studies, reported that when your favorite music is played, you see activation in your insular cortex. And if you're exposed to the music that you don't like, some other prefrontal cortex will be activated. So they do have distinct response. But for mice, I don't know. Yeah.
Great, thank you.
A question for Linda. So the subject that you tested, they were healthy subjects?
Yes, we tested normal hearing.
I'm just
adults.
wondering whether it would be interesting to look at mild cognitive impairment, because you talked about memory demand, and whether that could potentially lead to a potential biomarker. Look at those differences.
Absolutely. I think there are some really interesting applications for whether these two pitch systems may be differentially affected by different disease states. Potentially this memory effect could lead to a biomarker for mild cognitive impairment, and also potentially different forms of hearing loss will differentially affect these different pitch systems. So I think there's a broad scope to look at these as potential diagnostic markers. Yeah. Thank you.
One more question, but I want to let the audience and also go ahead if you have.
It's great to have so many engaged people online, so maybe I'll just throw in another question. This one's for Dr. Geffen. So would you say that there are two kinds of predictions in music? Sensory adaptation to remove expected sensory inputs, notes that are expected, versus feedback to sharpen expectations, which could then lead to a surprise?
Yeah, I think that's a really good interpretation. Of course, I think that's a really good interpretation of kind of our findings. I think that we haven't, of course, measured music directly. We used very simple sequences. We're trying to build up now and try to understand how second level order statistics are represented in the cortex and how that shapes these the adaptation to statistical structure of sounds and signals for a violation in the prediction.
Another question for Dr. McPherson. Trying to figure out whether it's F0 or their harmonics or the combination of the two that gives people a sense about whether the note is the same or different. I guess in my mind, I would have thought an experiment that would be looking at that directly would be to take a tone where you have F0 and the expected harmonics. And then another example where you keep F0 the same, but you move the harmonics so that it looks as if the tone went up. And then you ask people, did it go up or did it not? Have you done that one?
So sorry, maybe I don't understand. So you maybe present them with one harmonic and then present them with a set of different harmonics, or just shift the upper harmonics?
You keep F0 the same, but in the second note, you move the harmonics so they're no longer concordant with F0. You move them up, and you ask somebody, did the note change or not? If they're focused on F0, they'd say no. If the harmonics are influencing them, and there must be something about how prominent those are, they would say, yes, it went up.
Yes. So we've done some versions of that. So if you just have an experiment where you have the lowest frequency component, which is often called the fundamental frequency component, and then you shift to the upper harmonics, often the percept will then become that you actually just have two different notes present. So it's not that the entire pitch shift, it's that you actually just have two notes now, one that's higher than the other. What we can do, though, is actually sometimes pit the harmonics against the generative fundamental frequency. So we can take, say, harmonics 1, 2, and 3 and present that in the first note, and then present harmonics 4, 5, and 6 in the second note. And so both of those two notes can have the same fundamental frequency, even though the fundamental frequency component, the lowest harmonic, is only present in one of them. And you get this sort of sense of there's a shift up in that case because you have higher harmonics in one case and lower in the other. Yeah. And it seems like people can get a bit confused by that type of representation. So there definitely is a tension between whether people are estimating the generative parameter of this F0 versus whether people are perceiving the harmonic structure itself. So yes, there absolutely is a tension between those two.
Got it. Thank you.
Yeah.
Hi, thank you so much. Just, I guess, for all the panelists, just from the purpose of, someone asked an evolution question earlier, and I'm also curious because language has a use to communicate information, but music does not so much communicate information from one to the other. So I wonder if you have any ideas about how, like, why music might have evolved to, this is similar to the question, but how it might connect other regions that might relate to reward as well if it doesn't actually have a use like language does.
That's a loaded question, but thank you for that. That's exciting to think about it. I think people would argue that music often does convey information and can be used for communication. And in fact, something that I learned at the Music as Medicine Symposium is that it can be used therapeutically to facilitate communication with patients who have difficulty communicating using speech. So that's that's important to keep in mind. We communicate through music actually quite a bit if we start paying attention to it. But I do think that something that we did not discuss. So Kevin, touched on that, on the analgesic effects of music, but something that the kind of the pleasure and the reward signals that music builds on is probably underlies the evolutionary basis for it. There somehow these the predictability and the structure of melodies, or breaking that predictability, probably plays with the serotonin, with dopamine, with neuromodulation in our brain in such a way that can make music very pleasurable. It can also evoke emotional responses. It can help us heal. It can help us deal with complex memories, and also it brings people together. So even if we're not communicating by directly transmitting information through music, people can come together and share and digest experiences.
Yeah, and I should just say I play music too, so I fully appreciate music. But from an evolutionary perspective, I mean,
I understand.
Yeah, and so I guess just as a last follow-up, do you think music these are side effects of a system that was designed for something else, or is music a system in and of itself?
I think our brain has been wired to actually respond to music, and culturally, music actually is a connector. It brings social cohesion. So I think it's not something just that's outside of the realm of possibilities. That our brain really are really wired to respond to music, to actually anticipate the pleasure of music, so.
Thank you.
Francis, I don't know if you want to comment as a musician.
Can I ask a question for Dr. McPherson? So you're probably familiar with experiments by Daniel Presencer looking at shepherd tones, where context makes a difference, preceding context makes a difference in the perceived pitch change. So what do you think is going on there? Is that something where you're actually changing the spectrum? And another way of thinking about it is that these effects might be that FO is just a simpler sort of working memory load than trying to keep track of the whole spectrum. So could it be, this is a double-edged question, how do you explain the Presencer result in relation to your description of these different pitch systems? And also, how would you think about the memory aspect that's involved in an FO versus a full spectrum recall?
Okay, absolutely. So relating to the first question, I think that actually just dovetails into what we've been sort of discussing here about expectation. So I think our expectation can lead us to perceive, like I was mentioning in my answer to Dr. Collins, that if there is a tension between the harmonic structure and the F0 or the F0. And so if we are expecting one thing and we then sort of are presented with a potentially ambiguous stimulus like a shepherd tone, our expectations might play into effect there. I haven't looked at sequences like Daniel Presencer's work myself, so that's only speculation. But regarding to your second question about F0 and F0 and memory, I do think that this is related to the compressibility of the information. So these inharmonic stimuli are inherently uncompressible because they are random. The harmonics are randomly jittered in the way that we've done it. And so, however, with the harmonic tones that have a coherent F0, you can compress that into a single bit, a single number. And so I think that is one of the reasons that becomes an important factor for memory, is simply because it does allow you to compress a representation of the full spectrum into a single bit of information.
So I think we are at time, but I want to thank everybody for attending the symposium. If you're interested, if we have picture interest, please get in touch with us at music_box@mail.nih.gov. As we mentioned, we see some potential for utilizing music and other art form as therapeutic, having therapeutic benefits. But we need to better understand how music and other art forms exert their effects. And we feel that investigators that are involved in technology or tool and method development, particularly in the Brain Initiative, could be very, very helpful in collaborating with our neuroscientists that are currently doing work on music and health. So having said that, thanks again, and appreciate your attention. Thank you.